The lab that I'm going to demonstrate for you today is the RPR card test for syphilis. And this kit is made by the company ASI, the same company that makes the anti-streptolysin O kit that we previously performed in this lab. And RPR stands for Rapid Plasma Reagent, and this is an antibody that is produced in a person who has a syphilis infection. In this kit, there are 3 controls: non-reactive, weakly reactive, and reactive. This is the first time there will be 3 controls contained within a kit that are external controls we're going to add to the card during our testing. And it's also the first time that we are going to be listing our results or recording our results as non-reactive rather than negative, weakly reactive or reactive rather than just positive. Also in this kit, we have what's called the carbon reagent, and it's basically tiny little charcoal particles. And the reason we're using a charcoal particle rather than a latex particle or a red blood cell, as in previous kits, is because this is a flocculation test, not an agglutination test. And if you remember from lecture, a flocculation test is a special type of precipitation reaction. And so what's going to happen is antibodies, if they're present in the sample we're testing, will bind to the carbon particles in the carbon reagent, and we will see small clumps falling out of solution because those are the precipitates that are forming in the test. So I'm going to be performing all 3 controls and 1 patient serum sample today. I've labeled my card with non-reactive, so I'm going to add a drop of non-reactive control, a well-mixed drop of weakly reactive control to its reaction circle, and a drop of reactive control. Next, I'm going to add a drop of my patient sample, and the next step, which is different from other kits we've used, is that you spread the sample out using your dispenser to where the sample completely fills the reaction circle. So I'm doing that for patient sample, for the reactive control, weakly reactive control, and non-reactive control. Next, we take our charcoal reagent, our carbon reagent, and I've placed some of the reagent from the original bottle into this dropper bottle, which also comes with the kit, and it comes with a special needle that is going to put the proper size drop into the reaction circle so that we have a zone of equivalence if the antibodies are present, and we should see a reaction. This dropper, if you place 1 milliliter of reagent in the bottle, this dropper should make exactly 60 drops plus or minus 1, so somewhere between 59 and 61 drops. I previously QC'd this dropper, and it is delivering the proper sized drop. So what I'm going to do is mix up the carbon reagent and hold the bottle directly vertical and place 1 drop into the reaction circle for each of the tests that I'm performing. Alright, also in this kit, something that's different is that we are using a rotator. A rotator that will basically move the card around in a circle. And we'll do this for 8 minutes so that the reagents have a time to react. So I'm placing that on my rotator now. Okay. And setting a timer for 8 minutes. Now, as this kit, or as these reagents react in the circle, there is a special way that we are supposed to read these results. In order to see the small particles that are going to form, we are looking for either very reactive, larger particles forming, weakly reactive, a little bit smaller of a particle, also weakly reactive, very minimal particle formation, or non-reactive. Now, how we know that a test is non-reactive or there aren't any antibodies present is that we will see a little tail in the reaction circle, and that little tail tells us that there aren't clumps or precipitates forming. There are no particles actually forming. So this reading card is placed in each of the kits so that you have it to refer to while you're reading your results. Now, it's going to be hard for us to actually see this taking place on the rotator because I tried earlier, I can't get the camera to focus with it really rotating around. But after 8 minutes, you will read your reaction for the precipitates. And I'm just taking a small, quick look to see if I see any yet. You can't really see the reaction very well in the camera because they're so small, but it does look basically just like the picture. My reactive has the larger particles. My weak reactive has smaller particles, and my non-reactive is making a small tail. My patient is reactive. It's making pretty large precipitates already, so I know that it is positive, but I would allow the card to rotate for the full 8 minutes to make sure that I'm actually seeing a negative result for my non-reactive. It is really hard to see the tail in here. You can kind of see the precipitates forming in the other reactions now. A little better on the camera without moving the card around. So, with this test, there is also a semi-quantitative method. And if you remember from earlier in the labs that we performed with CRP specifically and RF, in order to do semi-quantitative, what we did is we took some patient sample and we created some, get rid of these, we created some 1 to 2 dilutions, and then from that, or a 2-fold serial dilution, and from that we tested the patient sample again. So what I've actually done here is I have labeled some tubes with 1 to 2, 1 to 4, 1 to 8, and 1 to 16 dilution. I have already added 50 microliters of saline because it tells you in the instructions to use saline for dilution. And now what I'm going to do is I'm just going to take 50 microliters of patient sample and make my first dilution. I'm going to make my 1 to 2. I'm going to mix it well, then transfer 50 microliters to the 1 to 4, mix, 1 to 8, mix, 1 to 16, mix, and now I can use these diluted samples to repeat the test as a serial dilution or a titer. So we're going to have a patient sample that's not diluted, a 1 to 1, a 1 to 2, 1 to 4, 1 to 8, and 1 to 16. And again, all we're going to do is we're going to take that patient sample, we're going to add a drop to the reaction circle. We already know that this patient has RPR antibodies. So what we're doing now is we're just trying to see semi-quantitatively how much antibody is in that patient sample. So we're going to perform the test just like we did before. We're adding our patient sample, spreading it out in the reaction circle to fill the entire circle, taking a well-mixed drop of carbon reagent, and placing it on our rotator for 8 minutes. And then as the rotator rotates the sample, we should get a positive reaction again in our undiluted patient. And then we will see if there is still a reaction in the 1 to 2, we look to the 1 to 4. If there is still a reaction in the 1 to 4, we look to the 1 to 8. And if there is still a reaction in the 1 to 8, we look to the 1 to 16. And we actually read this while it's on the rotator, but again, it's really hard to actually focus with the camera to show you that. And so I would simply let it rotate long enough to see what level the titer is at. I'm already seeing a reaction in undiluted, and in 1 to 2, and in 1 to 4, but not in the 1 to 8. So my titer stops at the 1 to 4 dilution, or a titer of 4. Now, if this person continued to make antibodies, that amount of antibodies would go up, and you would see reactions in the more dilute samples as that disease progresses. We will talk about the actual progression of syphilis in lecture in Unit 3.